Topic for today's lecture is necrosis and apoptosis. These are the two ways by which cell death occurs. These are our learning objectives that we'll be covering in this lecture. So, cell death is the end result of progressive cell injury. Whenever irreversible cell injury occurs, a cell dies. Cell death is important in the evolution of disease, and under physiological conditions, it occurs during embryogenesis, organ development, and to maintain homeostasis. There are two types of cell death, necrosis and apoptosis. Here you can see in this picture, the two types of cell death are shown. In case of apoptosis, the cell shrinks and later on the organelles, they are released into small blebs of the cytoplasm and then they are engulfed by macrophages. So as the cell does not rupture, no inflammation occurs. In cases of necrosis, the cells first swells up and because of this swelling, the organelles, they also swell up and later on the membranes, the cell membrane, the lysosomal membranes, and the mitochondrial membranes, they all rupture. So this leads to surrounding inflammation. So necrosis is a type of cell death that results from denaturation of intracellular proteins and enzymatic digestion of the injured cell. It is dependent on ATP, it requires energy, membrane integrity is lost during necrosis, and the surrounding tissue is inflamed. Common causes of necrosis are ischemia, hypoxia, toxins, infections, and trauma. Here, the morphology of a necrotic cell is seen. You can see the changes that occur in the cytoplasm include increased eosinophilia, and this is because of the loss of normal basophilia imparted by the RNA, and also because of binding of eosin to the denatured proteins. Then there is digestion of organelles, which results in cytoplasmic evacuation, and this is also called as moth-eaten appearance. The dead cells later on they are replaced by masses of damaged membranes. Then they are degraded into fatty acids because the membrane is made up of phospholipids. So when this is degraded, it turns into fatty acids, and later on the dead cell it is calcified. On electron microscopy, the necrotic cells are characterized by discontinuities in the plasma membrane and also the membranes of various organelles. There is marked dilation of mitochondria. Intracytoplasmic myelin figures are seen. Myelin figures are basically the remnants or the degraded products of the phospholipid bilayer membrane. There is amorphous debris of various organelles. Amorphous means they have no particular shape. There are aggregates of fluffy denatured protein, and there are nuclear changes due to DNA breakdown. Here you can see the changes that occur in the nucleus, and these are the patterns of nuclear changes seen in necrotic cell. These are karyolysis when the nuclear fades away, ecnosis when the nuclear shrinks, and karyolexis when there is nuclear fragmentation. And finally, the necrotic cell, it becomes anuclear. These are the various patterns of tissue necrosis, and they have been divided into different patterns because they are morphologically distinct and they provide a clue to the underlying disease. These are coagulative, liquefactive, gangrenous, caseous, fibrinoid, and fat necrosis. So, coagulative necrosis is a form of necrosis in which the architecture of dead tissues or necrotic cells is preserved, and this is because the denatured structural proteins and enzymatic proteins they block the proteolysis of dead cells. So because the enzymes are denatured, they are no longer able to digest the proteins. So the architecture of dead tissues is preserved. The cell is eosinophilic, anucleate, and then later on it is removed by phagocytosis. Common cause of coagulative necrosis is ischemia, which is caused by vessel obstruction, and coagulative necrosis occurs in all organs after ischemia except the brain, where liquefactive necrosis occurs. So liquefactive necrosis is characterized by enzymatic digestion of dead cells, and there the tissue transforms into a liquid mass. It is creamy yellow in color because of the presence of dead leukocytes, and this is called as pus. This is commonly seen in ischemia and hypoxia occurring in the central nervous system or in the neurons. It can also occur due to focal bacterial or fungal infections. Next is gangrenous necrosis, which is a form of coagulative necrosis, and it occurs in limbs that have lost their blood supply, or in cases of uh, uh, or in the gastrointestinal tract when it has lost its blood supply. It is called as gangrenous necrosis. When the gangrenous necrosis it is superimposed by bacterial infections, it transforms into liquefactive necrosis. And this is referred to as wet gangrene. Otherwise, it is called as dry gangrene. Next is caseous necrosis. Now, the term caseous means cheese-like and it is derived from the friable white appearance of the necrotic area. On microscopy, the necrotic area it appears as a structureless collection of fragmented cells and amorphous granular necrotic debris, which is basically central caseous necrosis, and this is enclosed by an inflammatory border comprising of epithelial cells, which are basically macrophages, lymphocytes, plasma cells, and Langerhans type of giant cells. So this collection of necrosis with inflammatory cells is found in granuloma. So this type of necrosis is usually seen in the foci of tuberculous infection. Now, fat necrosis, this occurs usually in cases of acute pancreatitis. In here, there are focal areas of fat destruction because during pancreatitis, there is pancreatic injury. So the lipases present inside the pancreas, they leak out and they split the triglyceride esters within the fat cells. So therefore, this leads to necrosis of the fat cells. So grossly, there will be chalky white areas as is seen in the picture above. And on microscopy, there are shadowy outlines of necrotic fat cells with basophilic calcium deposits, as it is seen in the picture that is below. Now, fibrinoid necrosis is a special form of necrosis which is seen in immune reactions involving the blood vessels. Because there is deposition of antigen antibody complexes within the walls of vessels, so this leads to deposition of fibrin or complexes are formed with the fibrin that leaks out from the damaged vessels. And this fibrin, it is bright pink in color and amorphous in appearance. So immune-mediated vasculitis, this shows fibrinoid necrosis. Now, what is the ultimate fate of these necrotic cells? They are either phagocytosed or they are calcified. Coming to the next form of cell death, it is apoptosis. This is named after the Greek designation for falling off, just like in autumn as the leaves fall off from a tree. In apoptosis, the cell organelles, they fall off from the cell and they are released in the form of small blebs. This is also called as programmed cell death. It is a tightly regulated program and it consists of intrinsic enzymes that degrade the cell. Later on, small apoptotic bodies are formed in which the organelles of the cell are present. The plasma membrane remains intact and therefore there is no inflammatory reaction in the surrounding tissue. There are various causes of apoptosis which may be physiological or pathological. Physiological as in during development and throughout adulthood. There are some organs that involute. Then there is deprivation of survival growth signals that also can lead to apoptosis. Apoptosis is also important in removing unwanted, aged, or harmful cells, like the webs of the fingers. They are removed because they are not required. Apoptosis also maintains a steady number of cell populations. So these are the functions of apoptosis. And cases of pathological, the disease cells and the damaged cells which cannot be repaired, they are removed by apoptosis. Physiological apoptosis occurs during embryogenesis, during organogenesis. So in fetal life, this is an important process. that regulates the organ development. Then there is involution of hormone-dependent tissues upon hormone withdrawal, like that occurs in cases of menopause, lactating breast after weaning, or when the food is started, when the oral food is started. So the cells of the breast, they also undergo apoptosis. Then prostatic atrophy, elimination of self-reactive lymphocytes, which are the body's own lymphocytes, and they are, because they are reactive to the body's own cells, they are eliminated. Then death of neutrophils that have served their purpose after they have cleared up the infection, they are also removed by apoptosis. And the intestinal epithelium, it is a rapidly proliferating epithelium, and there is rapid cell loss in these type of epitheliums. This is also because of apoptosis. Various conditions in which pathological apoptosis occurs are like in cases of radiation, when there is obstruction of duct in salivary glands, pancreatic ducts, when anti-cancer drugs are given, that also leads to apoptosis of the cancer cells and as well as the normal cells. Extreme heat can also lead to pathological apoptosis, and increased mitochondrial permeability is another factor that leads to pathological apoptosis. On morphology, the apoptotic cell shrinks in size. The organelles are more tightly packed. The cell looks like a round or oval mass of intensely eosinophilic cytoplasm. There is condensation of the chromatin. There is pycnosis. The nucleus eventually breaks up. Plasma membranes remain intact, and the organelles, they are released in the form of apoptotic bodies. In the picture, you can see an apoptotic cell in the epidermis. Now, regarding the mechanism of apoptosis, there is activation of enzymes called caspases. These are inactive coenzymes, and they undergo cleavage before they become active. There are two important phases of apoptosis: initiation phase, in which some caspases become catalytically active, and then there is the execution phase, in which there are other caspases which stimulate the degradation of cell components. In the initiation phase, there are two pathways, intrinsic for mitochondrial, and the other pathway is extrinsic for death receptor pathway. In the intrinsic pathway of apoptosis, the mitochondria is stimulated. There are some intracellular signals or intracellular microbes that cause damage to the mitochondria, and this causes release of the enzymes or the factors from the mitochondria, like the caspases are released, the cytochrome C is released. Cytochrome C, when it is released, it binds with procaspase 9, and this leads to activation of procaspase 9. It becomes caspase 9. Caspase 9 then activates caspase 3 and leads to apoptosis. Here in this picture, you can see a viable which is receiving growth signals, and therefore it does not undergo apoptosis. On the other hand, there is a picture of apoptosis when there is lack of growth signals or radiation or any kind of DNA damage. This stimulates the mitochondria to release its pro-apoptotic proteins and to release cytochrome C, and there is activation of caspases which leads to apoptosis. This picture is showing the extrinsic or death receptor pathway of apoptosis. Here, the mitochondria is not activated. Instead, the Fas ligand binds to Fas. This is the death receptor, and this activates the Fas death domain. This in turn activates procaspase 8, which turns into caspase 8. This is the active form, and then caspase 8 again activates caspase 3 and leads to apoptosis. So both the pathways, the intrinsic pathway and the extrinsic pathway, they converge on caspase 3. So this is the execution phase. In this picture, you can also again see the extrinsic pathway of apoptosis, which is activated by the death receptor. Now, execution phase of apoptosis is the part where the caspase 3 is activated and it leads to apoptosis. So both the pathways, the intrinsic pathway as well as the extrinsic pathway, to converge on the caspase. The apoptotic cells, they are released by phagocytosis. In apoptotic cell, there is phosphatidylserine serine expressed on the outer layer of the membrane, which makes these cells more palatable for the macrophages. Also, they are coated by antibodies and complement proteins, which helps macrophages to recognize these cells. Now coming to the clinical pathological correlations of apoptosis. As we've discussed, when there is growth factor deprivation, when the lymphocytes they are self-reacted or not stimulated, when the neurons they are deprived of growth factor, all these factors they lead to apoptosis. Then exposure cells to radiation or chemotherapy, this also induces apoptosis. P53 is a protein that can trigger apoptosis in cells that have damaged DNA. In tumors, what happens is P53 is absent, and therefore the cells which have damaged the DNA, they cannot be repaired or they cannot undergo apoptosis. They start proliferating and they turn into cancer. These are some of the disorders that are associated with defective apoptosis and therefore increased cell survival. These include cancer and autoimmune disorders. Disorders associated with increased cell death and excessive increased cell death are neurodegenerative diseases, ischemic injuries like myocardial infarction and stroke, and viral infections. Here is a picture that is showing the balance of living cells and dead cells. This maintains homeostasis. When cell death is extensive, this leads to neurodegenerative diseases, immune deficiency, and infertility. And in cases where there is less cell death, this leads to more proliferation of the cells, which means cancer and autoimmune diseases. This is a table showing differences between the two pathways of cell death. Necrosis is an accidental, unregulated form of cell death that results from injury, whereas apoptosis occurs when the cell's DNA or proteins are damaged beyond repair and the cell cannot repair itself, therefore it kills itself. Necrosis is always pathological. Apoptosis may be physiological or pathological. In necrosis, there is damage to membranes, which causes lysosomal enzymes to leak, while in apoptosis, there is nuclear dissolution, nuclear fragmentation, and the membrane integrity is not lost. In cases of necrosis, the cellular contents leak out, while in apoptosis, the cellular contents do not leak out. In necrosis, there is surrounding inflammation, while in apoptosis, there is no inflammation. Causes of necrosis include ischemia, hypoxia, toxins, infection, and trauma, while in apoptosis, it is a programmed cell death, so it is regulated by the cellular signaling. There are distinct signaling pathways for necrosis, which are different from those of apoptosis. Necroptosis is a form of cell death that shares aspects both of necrosis and apoptosis. It is a caspase-independent programmed necrosis. Morphologically and biochemically, it resembles necrosis, and it is triggered by genetically programmed signal transduction events. It is similar to the extrinsic form of apoptosis because there is activation of FAS ligand and there is receptor associated kinase activity. It is similar to necrosis because caspases are not activated. It is caspase independent and there is lysosomal leakage, there is increased production of free radicals, there is mitochondrial damage and ATP depletion. So necrosis is basically important in the formation of growth plate and cell death that occurs in hepatitis, pancreatitis, and neurodegenerative diseases. And also it is an important mechanism in post defense against certain viruses that have caspase inhibitors. This is all. Thank you.